I would like to thank the organizers for the invitation and giving the opportunity to tell you about how we use pheromones to control agricultural pests. Consumers are demanding safe, eco-friendly pest control for the food we eat. Without pest control, farmers would lose 30 to 80% of their crop, which threatens our food security. Crop protection can prevent $60 billion worth of crop loss just in the US and billions more in the world. To feed the growing population, farmers need safe, effective, eco-friendly pest control options. We turn to nature, which already has a solution: pheromones. Pheromones are signals used by many organisms and released from one member and affect development, physiology, and behavior of the members of the same species. They are used by many organisms, including fish, insects, mammals, to communicate. They are in the market, and they are safe, species-specific, safe for non-target organisms, effective at very low concentration. But their use has been limited to baiting traps and disrupting mating. At Pheronym, we think pheromones can do much more. I have been studying pheromones for more than 10 years. I identified the first mate-finding pheromone of C. elegans. C. elegans is a microscopic roundworm or nematode. It has a very short life cycle. It starts with egg, 4 larval stage, then becomes adult and lay eggs. It does all this in 3 days in laboratory conditions. So this was my postdoc training to identify mate-finding pheromone of C. elegans. My postdoctoral advisor said, this is a low-hanging fruit, and it will have a big scientific impact. However, there were contrasting opinions on whether C. elegans had a mate-finding pheromone. I was new to the field. I did not know at that time C. elegans was hermaphrodite. It did not need sexual reproduction. However, when it mated, it produced 900 offspring. If it didn't, it was only 300. So there was an advantage. I did the chemistry at the National High Magnetic Field Laboratory, and our collaborators at Caltech did the biology. And they used a mate-finding bioassay, and in this assay, they put a thin layer of bacteria, which is the food for C. elegans. They don't respond to any signal if they're hungry, so we had to provide them the food. Then they had 2 spots. One spot was the secretions I sent them. The other spot was control. And they put the nematodes, the spot that marked with X. Then they used a video tracking system to determine how long they spent in each spot. So as you can imagine, my first question was, when is mate-finding pheromone released? So I collected C. elegans secretions from all life stages and sent it to our collaborator, to Caltech. They tested both hermaphrodites and males. We thought hermaphrodites would be fairly indifferent or respond equally from secretions from all life stage, which it did, but males would respond differently. And males were interested in secretions from adolescents, young adults, and adults, which was consistent with their sexual maturity. So I selected young adults to collect secretions to identify mate-finding pheromone. After long and meticulous work, we identified the mate-finding pheromone 1.5 years later. It was not a low-hanging fruit, like my advisor said, it was on top of the tree. Identifying biologically active compounds, if they have multiple components, that is more difficult than identifying a single compound. And C. elegans mate-finding pheromone was a mixture of 3 different pheromones. So we published this work in Nature, and this work opened up pheromone world to the nematodes. You may be surprised, but nematodes are the most abundant animal on Earth. 4 out of 5 animals on Earth are nematodes. There are 15,000 known species, and many more have not been identified. They live everywhere from deep-sea sulfur vents to hot and dry desert climate. They parasitize nearly all mammals, insects, and plants. A species of Ascaris alone infect 25% of all humans in the world. And of course, they cause billions of dollars crop loss each year. The light patch, if you can differentiate on the screen, is a nematode-infected area. If that is not taken care of, it will get bigger and bigger and bigger. That means we are going to have crop loss every year, more and more and more. Because nematodes are so abundant and infect so many species, understanding how they communicate has a huge potential in medicine, veterinary medicine, and agriculture. As you can imagine, USDA Agricultural Research Service recognized that and hired me to apply pheromone technology to root knot nematode, which is a plant parasitic nematodes. These are the ones attack plants, and they cause more than $100 billion crop loss annually in the world. In insects, pheromones are used to interfere with mating and reproduction. So we could apply easily the same concept to the nematodes. We could interfere with their mating and reduce their population. Unfortunately, root knot nematodes do not reproduce sexually. So I was out of luck there. Luckily, pheromones actually control more behaviors, such as attraction, repulsion, sex, or dispersal in nematodes. This gives us the power to control nematodes. Just like bacteria, there are good nematodes and the bad nematodes. So we decided to bring our technology to market using the good nematodes. These are called beneficial nematodes. Beneficial nematodes are microscopic roundworms. They go and search and attack insects. They're commercially available, they're safe, non-toxic, target-specific, and leave no residue on our food. However, commercial nematodes do not work as well as they could. Pheronym has a solution. Dispersal behavior is one regulated by pheromones, and this is also important for effectiveness of beneficial nematodes. Here you can see an untreated plate. These are the nematodes that they lost the pheromone signal, and the one right in the middle, that blob, is full of nematodes. They're not searching, they're just staying in one particular location. And the other plate, we added pheromone. We call this product Nemastim. And now they're all searching that fuzzy little things moving around or wiggling normally. If we had a video, they would be wiggling. They're searching for an insect. So this is the base of our first product we call Nemastem. It's a patented technology. It provides safe and effective way to direct beneficial nematodes to search and attack. And the medium here is soil because we want to mimic what would they encounter in nature. And we place the nematodes, the untreated plate. And if you look at it, there is the insect away from the application site, is still alive. If you remember from the previous slide, they stay in one location, they're not searching. But when we treat them with Nemastem, they went and found the insect away from application site and killed. And it turned brown color there. And it is an additive for commercial nematodes. We mixed the Nemastem with nematodes. Now the activated nematodes are ready to spray to field for farmers. We tested other commercial nematodes, and it is broad spectrum, effective on commercially available beneficial nematodes, which control 28 of the most important pests. Another advantage is they are farmers do not need any additional equipment because they can use already existing equipment. Now, our next product in the pipeline is targeting plant parasitic nematodes, and we are first targeting root knot nematode. And it has a different mode of action, and this time we will be using seed coats. As Pheronym, we are unlocking the potential of pheromones for safe, eco-friendly pest control options. Thank you.